Okay, so I think we'll start the session. So I'll be sharing my screen. Someone please let me know whether the slide is visible and whether my voice is audible clearly or not. Anyone?
Audible, sir.
Okay. Thanks a lot. Perfectly fine. Okay, thanks a lot. Okay, so again, I welcome you all to the live tutorial session for week 5 of the course Bioinformatics Algorithms and Applications. The course instructed by Professor Michael Gomiha from IIT Madras. And again, as usual, like the last few weeks, so these Fridays tutorial session I will be taking. So I am Sunanta Samanto, a PhD scholar from IIT Indore at the Department of Biosciences and Biomedical Engineering. Okay. Now, throughout the last few weeks, we have covered some concepts, regarding the broad coverage of bioinformatics, sequence analysis, right? Sequence-based parameters, and then sequence alignment, phylogenetics, etc. Uh, so for these week's content, so for week 5, I think Professor Grumiha went over, like for example, some particular analysis of, sequence-based analysis of amino acid sequences, right? Stages of amino acid sequences where we calculated some like distances and then propensity, and then based on that, what the secondary structure formation and then content will be like, these kinds of things, and then some clustering algorithms, some information theory based concepts, right? So on and so forth. So we'll cover those concepts and then try to solve questions related to those concepts, which appeared in some previous iteration of the courses or similar to what you might find in your assignments and exam problems, right? Okay, so for just a brief coverage of the last week's class, so we found out that we could calculate, for example, in a weight matrix, the or any alignment matrix, uh, the value of certain components at certain position of the sequences, right? So which can tell us about the conservation or the propensity of that particular component to be present at a particular position in a sequence, basically. So position related information. So we found out that we can count, the overall occurrence for a particular position, and then we can convert that to some sort of log based values. So it can give us some sort of indication of like what the position based occurrence or what the position based uh, occurrence for the particular amino acid or particular nucleotide can be at a particular position, basically in a sequence or in a stage of sequences, right? And then what we did is that we went over the position specific scoring matrix again, further improving on that, like alignment matrix related concepts to give us a better understanding of the position specific scoring, which can tell us, uh, you know, the basically the occurrence of particular amino acids at particular positions. And again, here we did some counting, and then we converted that to frequency. Then we used the overall frequency and divided the sequence alignment frequencies to that with that overall frequency, and then we converted that log base 2. And then basically that could give us the position specific scoring matrices. Then we learned about the normalization techniques. Basically using formulas, we can normalize the stage of values between the 0 and 1, basically, right? And then ultimately we went over some concepts of phylogenetic tree construction. So we went over the UPGMA concept, where basically the distance between different sequences or different taxa, you can say, or different species. So we can use that, and then based on the lowest distance, we can basically combine two different species or two different taxa into one unit. Then you can assign some branch length based on the distances, and then after that grouping, again you can iterate through the process to ultimately get like an overall structure of the phylogenetic tree, where uh, basically you can find out which two sequences will be the closest in terms of evolutionary distance, and then their origin point or the diverging point, and then which other sequence the diverging point is connected to, and so on and so forth, right? And then we try to convert that using a simple uh, array of letters basically. So that was called the Newick notation for the trees. And then again, we did some conversion. We found out about rooted and unrooted trees. And then that was basically the overall concept about phylogenetic analysis kind of type. So today, before starting some concepts regarding the or solving some problems regarding the secondary structure, formation, information content, and clustering and so on, whatever was discussed in the classes. So let's recap a bit about. Okay, so let's recap a bit about the basically overall secondary structure formation, just a little bit sulfur helices and beta sheets and so on. Yeah. So basically, as we know, the types of secondary structures, one of the most prominent one are alpha helices, right? So the secondary structures are particularly formed based on uh, the arrangement in a way that they can form certain types of hydrogen bonding in a certain pattern, which can give rise to different sort of type of secondary structures, right? For example, here we have the alpha helices. Okay. So alpha helices, what they do is that the residues forming the helices, they have hydrogen bond formation between the hydrogen atom attached to electronegative nitrogen atom of a peptide linkage. And the hydrogen bond occurs with the carbonyl oxygen of the fourth amino acid on amino terminal site of the peptide bond. Basically, I and I plus 4 residues are forming the hydrogen bonds here. And then each successive turn of alpha helices is placed by 3 or 4 hydrogen bonds. And then these hydrogen bonds contribute to the stability of the overall alpha helical structures, right? So whenever the stability of a 3D structure, whenever that concept is coming, so there you have to know that secondary structures, formation of secondary structures essentially enhance the stability for a particular region. If that region is unstructured, you will see that that region will be highly flexible and it will be moving around in a solvated space a lot, right, in the physiological environment. Formation of secondary structures kind of are important to stabilize some part of that. So here again, alpha helix is doing the same thing. So if I ask one question, for example, I think we covered some of these in the earlier classes also. So for secondary structure, it is coming in the stability of uh, the secondary structure is basically providing the stability to the protein in a particular region. But for the folding of protein through the primary structure or the primary stage of amino acid into a higher order 3D structure. So do you remember what force or which interaction not really, but what kind of force was basically impacting or influencing that folding of protein in a solvated environment the most? Anybody remembers? So when the proteins are solvated, after that they will be stabilized by formation of hydrogen bonds, let's say van der Waals interactions, electrostatic interactions, and so on. But before forming these kind of interactions, so another force was important for the actual folding. And here I see one answer: hydrophobic interactions. Yeah, hydrophobic interactions or hydrophobic collapse is the region or is the reason that the proteins are basically folding into a 3D structure, tertiary structure in a solvated system. And that concept or that folding was primarily driven by entropy, right? So I think we might again visit that in the coming slides. So it was mainly an entropy driven process where based on the entropy and the disorder, the folding was basically occurring as a way to basically increase the disorderedness, which can happen if basically individual amino acids are or states of amino acids where there is a solvation layer around those amino acids. If they combine this highly ordered solvation layer, the size or surface or volume of that will decrease basically. So that will inadvertently uh, increase the disorderedness in a kind of way. You can think about it like that, right? So again, another type of secondary structure was beta strands. So it can happen in two types of arrangements. So these are for example, parallel beta sheets. So the strands can be ordered in this way, and then anti-parallel beta sheets also. So here beta sheets are basically occurring when the adjacent polypeptide chains they arrange either in a parallel or anti-parallel orientation. And for the parallel orientation, the chains have to be the same amino to carboxyl orientations, while in anti-parallel they have to be in opposite orientation. So here, if you notice, in case of the parallel beta sheets, for example, so the amino to carboxyl group. So this, if you notice throughout these chains, so different colors are highlighting your different atoms that are present here, right? So side chains are highlighted in purple here, and then for the red uh, color, so these red atoms, these are indicating your oxygens, right? And then the blue ones are indicating hydrogen, nitrogen, let's say, right? So here, if you compare the three different strands of the parallel beta sheets, you will notice that basically each same atom of these sheets. So if the blue atoms, for example, the nitrogens here, or the red atoms, or the R group side chains, etc. So they are particularly aligning just perfect in a perfect way, you can say, right? So these type of alignment will make that if you have to form bonding between let's say one nitrogen and one hydrogen, right? So that hydrogen bond, if it needs to be formed, then the bond will form kind of at an angle here, right? Like this one. In the case of antiparallel beta sheet, that is not the case, because here, if you notice, uh, that the strands are not aligning kind of in a perfect way, but it is sort of skewed to the side basically, which makes one blue atom here and then one red atom here. Those to be fall in a perfectly straight line, just one below another. You can think of it like that, and then that basically forms a kind of straight kind of hydrogen bond. So that is the particular bonding pattern that we will notice because of the arrangement of the amino acids and the positioning for the beta strands that compose the parallel or anti-parallel beta sheets. So they have, of course, somewhat similar structure, and their repeat period is slightly different, because for parallel conformation is 6.5 angstrom, and then for anti-parallel it's 7 angstrom. Now, as I said, hydrogen bonding pattern is slightly different. So interstrand hydrogen bonding are in line for anti-parallel beta sheets. This can only happen because the exact same atoms are not kind of perfectly in line, but it is slightly skewed, which makes the hydrogen bonding forming atoms, so one nitrogen and one oxygen, let's say, those to be perfectly present in line with one another, right? So that gives rise to the anti-parallel sort of conformation. And then in contrast, of course, in the parallel variant, the hydrogen bonds may be distorted or not in line. So here, because let's say the exact same atoms are present just one below another, so in line, so that means that the hydrogen bonding forming atoms, which will be nitrogen and oxygen, different atoms, so they will not be present in line. So the bond is kind of forming at an angle like this, which gives rise to the parallel beta sheet formations, right? Now, so for the parallel beta sheets and the anti-parallel beta sheets and the alpha helices and any kind of secondary structures, we discussed about that they have certain ideal phi and psi angle values, right? So for example, for alpha helices here, the values are given for every beta sheets and the beta turns, etc. The values are given. They will of course be present not as the exact value, but let's say in a range. But overall, this can be the ideal phi and psi for the secondary structures. And then based on that, we kind of discussed, I think, in an earlier classes. So what is happening is, because of course, okay, so first of all, the phi and psi angles are mentioned here. So this n and c alpha, the rotation between those will be phi, then the c alpha and c, that will be psi, etc. Right? And then because for every beta sheets, alpha helices, and every kind of secondary structures, so due to their particular sort of orientations, there will be certain constraints on the orientation of their side chains also, right? So it's not that every single phi and psi angle that is forming between different residues. So it's not that the value of those angles can be anything. But because of the particular orientation of side chains, that is sort of being restricted by their side chains, of course, and then the atoms forming the phi and psi angles, etc. So because the overall pattern of the secondary structures, so the phi and psi angle and their orientations of the forming atoms basically will be restricted uh, to minimize the steric hindrance. And here throughout these phi versus psi plots, so these are Ramachandran plots, right? Here we have let's say the range or the region where if you see these kind of values, so this could indicate that okay, so these residues are forming this type of secondary structures. For example, here we have parallel beta sheets, anti-parallel beta sheets, then this is left-handed alpha helices, right-handed alpha helices, and so on. And let's say that you do not know the uh, structure or the 3D structure of a protein beforehand. So we'll learn in some of the later classes that we can actually use different algorithms or modeling techniques. For example, homology modeling, or right now, for example, we can take the help of some of the AI-driven structure prediction algorithms like AlphaFold and so on. So those will give us, as the 3D structure, the coordinates of the atoms, right? They will predict those based on different types of algorithms. It could be homology modeling related, or it could be again some trained AI algorithm basically. But after they predict or model a structure, then you have to kind of go for these sort of analysis where you for the let's say entire stretch of amino acids forming that protein, where you find out in the given 3D structure where you find out the phi versus psi values for that protein, and you kind of plot each amino acid residue there in this phi versus psi plot, and then you see whether you are seeing any sort of unexpected distribution of the phi and psi angles for certain residues, right? So based on that, if you have a particularly good phi versus psi plot in the Ramachandran plot, so where you are not seeing deviation for residues, you know, outside these regions, outside these allowed regions, basically, then you have to kind of sort of go for the modeling again, or you have to sort of get to some minimization algorithms and then try to minimize the structures to optimize their phi and psi angle values for the amino acid residues there. So you have to kind of go for this Ramachandran plot analysis in a newly modeled structures as well. So that is kind of important because that will form the basis of your further in silico calculations, or let's say you are trying molecular docking or molecular dynamic simulations and so on. So you have to make sure that your initial structure that you're starting with is actually well optimized, right? So that's why you have to sort of do these kinds of analysis to fine tune those predicted structures as well. So that's why these are important essentially, right? Now, I think this question I went over in the earlier classes as well. So can somebody mention very quickly what could be the answer of this one? For the following options that are given, so which of them is not true about secondary structure of proteins? So which of them is not true? Which of these is not true? So can somebody quickly mention, or unmute or type in the chat? Yeah, I think I see one answer, answer A in the chat, right? So it should be A because as we discussed as well, uh, the hydrophilic slash hydrophobic characteristics of amino acids, those generally come into play when forming the fold of the protein, the 3D fold or the tertiary fold of the protein, basically. The secondary structures are important in stabilizing those folds, but the primary driving force for the folds are hydrophilic slash hydrophobic, which do not essentially come up as the most important thing in the secondary structure formations, right? Because secondary structures are, of course, formed due to these intramolecular hydrogen bonds that are kind of giving rise to the structures, right? Particular patterns. And then here also alpha helices, beta pleated sheets, and beta turns are of course the examples of protein secondary structures. So here the answer would have been A, of course, right? Uh, so here we just discussed this question. So for the clustering of hydrophobic amino acids, that was driven by entropy in a solvated system, right? So this was happening because of the solvation layer that is present around the individual amino acids, or let's say the stage of the amino acids. If it was like this, right? So a solvation layer is basically present around these each of the individual amino acids, and the solvation layer basically happens because when you are dropping an amino acid stage in water, so the water, because you are kind of disrupting the already present hydrogen bonding between the between the water molecules there, so those will the water basically will tend to gather around these individual amino acids, and then they will form a solvation layer around them. Now these solvation layers, the important thing is that these are very, very highly ordered structures, the solvation layers that are forming around the individual amino acids. So because they are highly ordered structures, so there will tend to be some sort of change or some sort of, let's say, reaction. It's not exactly chemical reaction. It's like a mechanism. So there will tend to be one mechanism which will try to increase the disorderedness, right? And for increasing the disorderedness, what will need to happen is that, let's say, if the surface area or the volume covered by these amino acids in total, if that decreases, then as a consequence, of course, you will kind of increase the disorderedness, because the volume covered by, or the surface area covered by these solvation layers, that will give rise to highly ordered structures, right? That is giving rise to the highly ordered structures. So what will happen is that these amino acids will tend to clump together, because when they tend to clump together at a fold, if you compare their overall, let's say, solvation layer that is formed around them, then the volume of this one will be lower, or the surface area covered by this solvation, this layer that is formed around the clumped amino acids together, that will be lower than the total sum of the total area of solvation layer that is formed between these individual amino acids. So because of this lower surface area covered or lower volume covered, what will happen is that the surface area or volume that is corresponding to the highly ordered structure, that will decrease, and as a consequence it will increase the disorderedness. And so basically due to these entropy related optimization, you can say, in a way, so that will give rise to the fold, basically, right? Fold or 3D structures here. So it is mainly driven by a favorable increase in entropy. So the clumping basically increases the entropy of the system in this case. Okay. So in the current week's class, Professor Grumiha went over some concepts regarding dataset construction and the importance of introducing or reducing the bias in the data, basically making the data more non-redundant in a way, right? Your dataset that you're analyzing. So can somebody quickly mention what does non-redundancy mean in this case? So if you are analyzing, let's say, a data set, or a, let's say, set of sequences, right? You want to analyze. So here it is mentioned that the data set has to be non-redundant. So can somebody mention quickly what does non-redundant mean in this case? So you can type in the chat. Yeah, so I see one answer. It is not similar. Exactly. So because if you take similar data, you might introduce bias in your analysis, right? So you look at it. Uh, so the example that Professor Tomiha went over in his lectures, I will cover that. So let's say here I have these two sequences, right? Let's say this is sequence 1, this is sequence 2. Now, what we want to calculate is, let's say, the composition of alanine in these two sequences, right? Or not individually here. So as a total, if you consider these two sequences, So what will be the composition of alanine? Let's say in a kind of sense of frequency, right? So can somebody calculate and mention? So let's say as kind of a frequency kind of way. So you have to first count the total number of alanine, then divide that by the total number of amino acids. So can somebody quickly mention in the chat? Uh, so not individually. I think I see one answer in the chat, seq1 0.3, but as a combined, as a whole, combining these two sequences together. So think of it in a way that you have a lot of sequences, and from that you are trying to infer, okay, is alanine more prominent in this data set, or is some other amino acid more prominent in this data set in that way? So you have to kind of combine both of these sequences together as a whole, and then compute the frequency or the occurrence of alanine. So here, for example, if you see how many alanines are present, in sequence 1, you will see 1, 2, and then 3 alanines, right? And then in sequence 2, of course, you are seeing 1 alanine. And in total, the sequence 1 and sequence 2 combined, the composition of alanine in this data set will be 4 by 20. So that will be 0.20, right? Now, what happens if we introduce in this data set another sort of data point or another sequence that could be very similar to any one of these two, the sequences that are already present in the data set? So let's try that one. So let's say this was our sequence 1. So this is our sequence 2. And now here what we are doing is that we are introducing a new sequence, which is sequence 3 in this data set. And now let's say we are trying to calculate the composition of alanine again. So can somebody mention? So sequence 1, 2, 3 and combined in this new data set where you have added sequence through as an additional sequence here. So what will be the composition of alanine? You can try to calculate and type in the chat. So I see one answer, I think 0.23, 0.23 again. Okay. So basically, how many alanines are present? So in seq1, we see 1, 2, 3 alanines. In seq3, we'll see 1, 2, 3 alanines. And in sequence 2, we see 1 alanine, right? So total 7 alanines. Divide that by 30. The total length of the sequence is combined, right? And then you get a value of 0.23, right? Fine. Now let's say we are, instead of introducing sequence 3, which is very similar to sequence 1 here, as you can see, let's say we are introducing something that is not similar to sequence 1, but instead similar to sequence 2, right? So let's say in this case, so this was our original seq1, this was our original seq2, yeah, I think, and then this, let's say we are introducing a new sequence, sequence 3, and then if I ask you again to calculate for this new data set, this new sequence, sequence 60 combined together, what could be the composition of alanine? Can somebody calculate and mention again in the chat? Yeah, so I see a couple of answers. 0.16, right? Yeah, so basically how many seq1, how many alanines you have in total? 1, 2, 3, I think I see here, and then 1, 4 here, and then another 5 here, right? So it will be 5 by 30, which will be 0.17. Okay. Now from this example, you see that if we are introducing, let's say, similar sort of similar type of data points that is already present in this data set. In this case, we have done it 2 times, slightly differently each time. We are seeing that it can basically bias the overall calculation of the alanine composition. Because if we are, let's say, introducing another sequence which is very similar to an alanine prominent sequence, very similar to a already present sequence in the data set, we can see that the overall composition of alanine will increase slightly, right? And then again, if you are introducing another sequence which is already kind of present, but not the exact same sequence, but very similar sequence, highly identical sequence, then that can again underestimate the composition of alanine in a way, right? Where if in the newly introduced sequence, the alanine prominence was not that high. So now let's say you have a data set, and let's say you have data set for a particular class of proteins, right? And then you are trying to identify, okay, so for this class of proteins or for this type of proteins, so what, I want to infer some sort of functional characteristics or features of these proteins, right? So you try to calculate, okay, firstly, I will go through the composition, right? Now, in this whole data set, let's say you have included certain data set or certain sequences which are very similar to one another. Let's say they have a very high sequence identity with one another. They are different proteins, certainly there is some differences, but they have a very high sequence identity with one another. Let's say in your total data set of 1,000 sequences, right? You have, let's say, I say 60 sequences which have greater than 90, or let's say, to make it more clear, we'll say out of these 1,000 sequences, let's say you have 60 sequences which are having greater than 90% sequence identity. Okay. So now you can imagine that if you are trying to infer from the composition analysis that what different types of amino acids are present in this sequence or not, or you are trying to decipher some sort of functional interpretation from that. So you will see that you will most definitely skew your results, whatever output you are getting from your analysis, right? Because you have included 60 sequences that are very similar. So it is possible that you will not exactly represent, okay, what your outcome would have been if you had sampled it in a way where similar sequences was not present in your dataset, basically, right? So these can basically skew your analysis results or outputs and can give you some sort of wrong inference or information. Basically, the same thing if you are trying to imagine, let's say, in a more simple sense, that if you are using, let's say, the same sequence 10 times in a set of 30 sequences, you are putting the same sequence 10 times. So, of course, that will, differently estimate the calculation or the composition of the amino acids, right? So, that can give you wrong information or wrong interpretation. So that is why it is important that whenever you are trying to do some analysis, let's say in this case sequence analysis, based on, of course, what you want to achieve from the calculations. So you might need to reduce the redundancy in your data, basically, right? Now, reducing redundancy, of course, in this case, reduces the bias that can occur in your data set, right? Which can give you more accurate results that can accurately represent your data set. Okay. So some of the softwares or programs that Professor Grumiha, I think, went over that can help you, uh, you know, reduce the redundancy in your data set. So can somebody give examples? What kind of softwares Professor Grumiha went over or mentioned in his lectures? Softwares that can help you, basically, given the input of a data set and setting some sort of parameters. Based on those parameters, the softwares can help you reduce the redundancy in your data set. So one of the softwares Professor Gromiha went over was this one called CD HIT, right? So it stands for Cluster Database uh, at High Identity with Tolerance, right? Now the concept of these softwares or this particular program is basically, so it uses something called greedy incremental clustering. So firstly, what it does is that, so you are providing, let's say several sequences, let's say 20 sequences. What it will do is that, firstly, it will sort the sequences by decreasing order of length of the sequences. Okay, so firstly that sorting is coming, and then what it will do is that it will take the longest sequence, whatever was the longest one, and that sequence it will consider as the representative of a cluster, which will basically, so cluster essentially means a grouping of data points, right? So in this case, the first cluster, the representative of the first cluster, the first component of that will be the longest sequence. Now, after that, what it will do is that whatever sequences you have remaining, so those are now compared to the existing cluster representative. So let's say you are comparing now the second sequence, and then you already are considering the let's say longest sequence as the first cluster, right? Now the second sequence, what it will do is that it will compare that with your first cluster representative, the first sequence in this case, and then based on some sort of threshold, let's say similarity threshold, identity threshold, or something like that. So based on that, the program will decide, okay, should this new sequence be a part of the already existing cluster, or should this be considered as a new cluster representative? Okay, and then based on that similarity threshold, that is as it is written here, the sequence either, of course, joins a cluster or a new cluster is formed, and then this process is iterated for all of the sequences that are present there, and ultimately you will see that definitely if you have certain sequences that are very highly similar with one another, so those will be grouped into one single cluster. Basically, let's say this cluster has components, these ones, and then let's say other clusters are having like this one, let's say has these components, and then let's say these clusters are having independent components. There is one, there is one, there is one, and so on. So if you consider, let's say, each of these single circles here as a cluster, then you will see the dotted data points or the sequences will be representatives of that cluster, which will be derived from like this kind of sequential way. So first the longest sequence, and then after that comparison of the other sequences with this longest sequence to find out whether they should be considered a new cluster or a component of the already existing cluster. Now, of course, here what I mentioned in that, so you have to compare two sequences basically, right? To find out whether they have high similarity or not. So the thing that you need to do is that you need to do some sort of alignments, right? Or somehow you have to find out the similarity between these clusters or between two sequences, right? Now, if you try to do, let's say, an alignment, so the alignment algorithm will take some time to run basically. Now, if you have a large data set and for each new sequences, you are trying to align those with the previously present representative of the clusters, and then you are trying to see, okay, based on the alignment scores, whether they can be new or the already remaining cluster part. So the exact alignment procedures will take longer. Now, to circumvent that, what these CD-HIT program uses, it uses something called a short word filter. Okay. So now what this short word filter essentially means is that, let's say you have uh, two sequences. If I give you an example, let's say just try some random protein sequence. Okay, A, G, T, R, L, G, Y, M, A, S, L, something like that, right? Some random stretch of sequences. Now let's say we are again getting another sequence. let's say this is our seq1, what I have written here, and then this is our seq2, right? Now let's say one of them, seq1 for example, was the representative of an existing cluster, and now we need to find out whether this new one, the seq2, whether this is a new cluster component or part of an already existing cluster based on the similarity. So of course you can do alignment, but if you notice, if two sequences are having a high degree of similarity or high, let's say, identity, so you'll notice something, that not only their overall, let's say throughout the entire sequence length, not only they will match, but you'll notice that if you sort of take small stretches of subsequences, for example, this AG here, so you'll find another corresponding subsequence matching on the other one. So AG will be present here again, TR will be present, so R is not matching, of course. So let me just erase that. Yeah. So AG, let's say a matching subsequence that is present in both cases. Let's say LV was matching, right? MA was matching, AS was matching, SL was matching. Here you can say GT was matching as well. So you will see that if you compare to large stretch of sequences, we will find short stretch of characters, small stretches of characters. It could be, again, as we discussed in some of our earlier classes, like dipeptide steps. So 2-character match, or tripeptide, not peptide. So 3 amino acid stretches, 2 amino acid stretches, and so on. So basically 3 word or 2 word stretches or characters you can think that will be matching. This has to be, there has to be some sort of number of matches like this for using small words or small subsequences if the two sequences have to be present at a high level of identity, right? So based on these kind of information, so what these the program does is that it uses that as a filter process. So rather than exactly aligning the two sequences, it tries to find out whether it has stretches of small subsequences or words, let's say in this case. So stretches of small words that are matching in both of the sequences, and based on those calculations, it will tell, okay, whether this will be a part of this cluster or not. Because of course, if you have a high number of, let's say, 2-character matches or 2-word matches in these two sequences, you can say, okay, so the overall sequences will be aligned in a good way, right? So it will be part of the same cluster essentially. So there are actually some statistics regarding these calculations. So let's say if two sequences, let's say 100 amino acid stretch sequences have 80% identity, you will find out that these two sequences will have 70 identical dipeptides. 70 identical dipeptides. You will find out they will have 55 identical tripeptides. We will find out they will have 25 identical pentapeptides and so on. Okay. So for 80%, 85% identity, you are expected to find these sort of statistics here: 70 identical dipeptides in these two sequences, 55 identical tripeptides, 25 identical pentapeptides, and so on. So based on that, based on this sort of property of identical sequences to have stretches of characters, stretches of matching characters to be present in both of them. So based on that, let's say you are providing a threshold based on that. So using those thresholds, it will find out identical dipeptide, tripeptide, etc. For those sequences to be considered similar, right? And then what it does is that it limits apply based on the clustering threshold. So here, of course, there are some limitations. For example, 70% similarity for pentapeptides, and then you can do statistical analysis using the short word sequences to find out basically the whether they are part of the same cluster or not. Now there are some limitations here, as it is said here. So sorry. So short word filter cannot be applied below specific clustering thresholds. So here, for example, they have theoretical minimum for thresholds is 80% for pentapeptide, 75% for tripeptide, and 65% for tetra, 65 for tripeptide, 50 for dipeptide, and so on. So what this means is basically, let's say in the worst case scenario, if you have two sequences which have 80% identity, right? In the worst case scenario, what it can happen is that the mismatches that you'll notice in those two sequences. So the mismatches that are present, they will be distributed in a random way, or they will be distributed evenly. Let's say, so they will be distributed evenly throughout the stretch of the length of the sequences. Perhaps the clearest one you can sort of get from this example, the example D here. So protein A and protein B, they have 50% identity, let's say, right? Now that means that 50% are not matching, right? 50% of the characters present are not matching, right? Now, even if, let's say, in a normal circumstance, even if they are not matching 50% of the time, what it can happen is that, let's say in a normal circumstances, that those 50% will be present like as a group in some way or in some part, right? In this cross, let's say these are mismatches, and then let's say some matches, some mismatches, and so on, right? And so on. So these can happen in a normal circumstances where these regions will have matches, and then these crosses will be mismatches, and then again some matches, some mismatches, some matches, and some mismatches, and so on, right? So in a normal circumstances, this will happen. You will notice that in this case, let's say some of the mismatches are grouped together, right? So they are kind of present adjacent to one another. So that will mean that, of course, there will be some regions where matches will be found. So a stretch of matches will be found, and in those regions, basically, you can compare the words and say, okay, these are matching together. But in the worst case scenario, if your 50% match was distributed or mismatch, let's say, was distributed evenly throughout the entire stretch of the sequences, for example, here, so the first M versus M, this one is matching, then the V versus T obviously is not matching, so there is a cross here, then G versus G is matching, then the next one, D versus E is mismatched, then H is matched, right? You'll notice that even though they have 50% matches or identity, you will not find any dipeptide sequence that is the same in these two proteins, right? In these two protein sequences, because the mismatches are distributed evenly. So you'll not find a stretch of sequences. In this case, you cannot even find a dipeptide, let's say, which uh, basically will indicate that the same characters are present in both of these sequences because the mismatches are distributed evenly. So the same thing if you apply at like a higher identity here, for example, in this case, in the first one, protein figure A, protein A versus protein B, 80% identity. But if the mismatches, so the other 20%, which will be mismatches, so if those 20% mismatches are distributed evenly, we'll notice that, for example, here, you'll notice that the first four characters are matching and then the next one is mismatched, right? The next four are matching, let's say, and then the following one is mismatched. You will notice that you will not be able to compare these two sequences in figure A using a stretch of 5 characters. So you cannot use a 5-word filter because the mismatches are distributed in a way that you will not find a 5-word match between these two sequences, right? So based on a 5-word filter, you cannot reduce the data set if in the worst case scenario the 20% mismatches are distributed evenly. So that will mean that to compare these two, you will have to use a shorter stretch of characters, so shorter words to match between those. For example, if you use 3-word matches, so you will find that 3 words are matching a lot of time, right? Here they are matching, here they are matching, here they are matching, and so on. So 3-word stretch you can use to calculate the matches or the mismatches, but 5-word filter you cannot apply here because you will not find a 5-word match anyway. So those are some of the limitations here. So for pentapeptide matches, you have a threshold of 80%. So basically, if you have, let's say, a lower degree of identity than 80%, so basically a higher level of mismatches, then you should not use that for that matching or clustering. You should not use a pentapeptide or 5-word filter in that sense. Then similarly for 75%, that is the threshold for tetrapeptide. 66.67 for tripeptide, 50% for dipeptide, and so on, right? So based on these probabilities, right? Probabilities in the case of event distribution, essentially. So in the slide, I have basically what I have done is I have given two links which you can maybe take a look at later. So these are the links for the software, right? And some of the user guides that are mentioned for CD hit particularly. So if we take a quick look at an example, okay. So hopefully you can notice another terminal that I have opened here. Yeah, I think you can notice here. So here, what we'll try to do. So first of all, if I let's say if I use okay, here I think I have already opened up. Yeah. So from the CD hit, you can basically download from these the website for the program. So from these paper on Oxford Academic Bioinformatics, the link is given for the software. I think it was yeah. Here it is available on this web page, and then you can download it from here, or alternatively you can do what you can do is that you can use Conda to install CD hit basically, right? So if you have Conda on your system, let's say you're running on a Linux system, so you can use this Conda install PyConda ::, CD hit, so it will install the CD hit software, right? Okay. So hopefully, please let me know whether this terminal is visible or not. So are you guys able to see this sort of maroon color terminal? Yeah, I think those are visible, right? Anyway, so what we will do is that I have already installed the CD hit software here. So just as a quick demonstration, so I have one data file called input.dat, and what I have basically done here is that I have taken the FASTA sequences for certain I have taken the FASTA sequences for certain amino acid states, right, or certain proteins here. Okay, so we'll see, for example, here the first one is human tyrosine kinase JAK1, then cyclin dependent kinase, then human isoform of cyclin dependent 2 kinase, etc. So these seem to be some kinase proteins for those I have for which I have taken some sequences, right? So total of let's see how many number of sequences are present here. So here we have the first one, second one, the third, 9, 10. So it seems that there are total like 19 number of different sequences in FASTA format in this data file, right? Now what I can do is that I can use this command that is given here. So cd hit, which is the name of the program using this Linux terminal, -i. So -i in a lot of command line, in a lot of command line syntax, basically it includes indicates that after that you have to provide some sort of input file. So here I will have the input file input.dat, and then -o will be the output file output.dat. And then I have some parameters here. For example, -c 0.9, -n5, -m8000, -t4. Okay. So these parameters basically indicate your provided basically parameters which will be considered by city hit. For example, these ones, the last two, so minus m 8000 and minus t4, those are basically the minus m is basically your memory limitation that you are giving for these software to run. So it has a memory limitation, let's say of 8000 MB, and then minus t is how many cores of CPU I will use to run this program. Then you have minus c 0.9 and minus m5. So these two are basically important parameters to give for CD hit. So this minus c essentially indicates a threshold value, a identity threshold of 0.9 or 90% in this case, that will be right. And then following that we have minus n5. So this minus n will indicate how many characters for the words you want to use for matches, right? So we discussed short word filter. So it will be minus n5. That means it will use a 5-word filter essentially. Okay. So if I basically let's say copy this command and then if I basically paste it here, if I run it on my computer, so what will happen is that because I have a much lower number of sequences in the input, so it will run very fast. I am using 4 cores, of course, 4 CPU cores, and it will give you some sort of output on the terminal. So what it says that some sort of parameters are being displayed here, which is indicating how your program executed, the software executed, and then as the output file, you will notice that this output.dat and this output.dat.cluster, these files are given. So if you open up the output.dat here, the output file for CD hit, you will notice that the total number of sequences, if you count, you will notice that these sequences have decreased. The total number of sequences have decreased in the output.dat. So basically what is happening is that based on the word filter and based on the provided identity, CD hit has considered if it has considered particular sequences to be duplicate or similar as another sequence, so based on this clustering, so it has excluded that from the dataset. You will get the information about that in the output.dat.cluster file here. If you just quickly open up, you will see that from 0 to 16, so a total number of 17 clusters CD hit has formed in its run period, and you will notice that in this cluster 6, there are 3 sequences 0, 1, and 2, which are kind of grouped together as cluster 6, right? And the information you will notice that this FASTA format one, which has this accession code P43405, it was basically this KSOIK underscore human. So basically a human SOIK protein, you can say, right? And the other 2 sequences which are grouped together, those were, if you notice from this visible part that is present here, so you'll notice that those were the SOIK sequences for mouse and rat. Okay, and those were basically at over 90% identity. So at 92.53% and at 91.57% with the KSYK humans. So CD hit has considered those as part of a single cluster. And in your output file, you will notice that it will only consider one of those. So the representative sequence from this cluster it will consider. I think it will be KSYK human in this case. Okay. And for the rest of the clusters, only single component or single sequence was calculated to be part of those clusters. So in total, from 19 sequences that we had, so 3 sequences are grouped together here in cluster 6. And then in the output file, we are getting essentially those 2 extra sequences are excluded. And then we are getting a total number of 17 sequences. That should be in the output file. So that is just a short demonstration of how you can use CD hit. So you can use this command line parameters. You can give the parameters through command line and you can basically get an output file with your provided set of word filter and identity threshold, right? Okay, so that was just very quickly about CD hit. I think, okay. Wait, so I think, was it visible or?
Hello.
Yeah, please.
It was not visible. Actually, I understood what you are trying to do and what you did it, but it was not visible. So, can we just have a magnified look at what was the whole thing for
Uh, Magnified means so by
zoom in
okay, I don't
because
see.
it was not at all visible. I could understand what you were trying to do and how the clusters were being formed. What was the result?
Mhm.
But
Okay,
what was written over there, what this we couldn't, I
okay,
couldn't
okay.
see it. It is very,
Bloody.
it
Okay,
is
okay.
not at all visible clearly.
I think because the font size is smaller, so you are able to see
Yeah.
the maroon terminal, but you are not able to read whatever
Yes.
is this. Okay.
Yes. Yes. Yes.
Okay, okay. Just a moment. I think what I will do is that I will just, let's say, I will just one. So I think I'll just capture the screen here and then show you this one. Okay. Let me just share my other screen here. Okay, so hopefully in the screen capture you can see. I'm trying to zoom it.
Yes. 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 We can see now.
Yes, yes. So what is happening is that so in this terminal I had basically some files. Uh, this input.dat. So this input.dat file is where I basically put 19 sequences, 19 kinase protein sequences in FASTA format. Okay. And then after that, using cd hit, what I did was that so I gave it the command which is written here in this terminal. So cd hit -i, then input.dat, and then -o output.dat, then -c. I provided a threshold for identities, so it will be 90%. Then -n, I used the word filter size, so it will be 5-word filter. And then after these two you can kind of neglect. So the M is the memory limitation for this program to run, and then T is how many CPU cores I want the program to use while running, right? And after it has finished run, because there was only 19 sequences, so it will take like a couple of seconds, right? So it will be very fast. After that, you will notice this sort of thing was basically given in the terminal. So here the program is basically cd hit, and while executing, some sort of parameters are mentioned here. So for example, total seq is 19, longest and shortest your length of the sequences was 1198 and 264. So we mentioned these that it will first arrange the sequences in a order of length, right? And then it will consider the longest to be the component of or the representative of the first cluster and so on. Then it was total letter and something like that. And then some other parameters are mentioned here in this terminal. So here you will notice that in the output, while after the execution of the command, uh, the program basically pasted on the terminal that 19 sequences it has finished running, and it has found 17 clusters, right? So 17 clusters, meaning if we, let's say, if we just take another screenshot and then open it up. So you'll notice that the 17 clusters, you'll notice that it has ranked from cluster 0 to 16. And in cluster 6, you will see that it has grouped 3 sequences together. And those are basically the SYK protein for humans, mouses, and rat. Of course, you will expect that they have a high level of match, right? So based on that, it has clumped these 3 together. and overall it has given 70 total clusters basically. And in the output file, you will notice that these three will not be present, but only one from this representative cluster will be present. So that's all, basically.
Thank you so much.
Okay, okay,
Thank
fine,
you.
fine. Let me just stop presenting here and then start the other screen. Okay. Yeah. So that was basically it for CD hit. And then let's say, okay, yeah. So these are some other information that are present here. Uh, so you can see here that it has, of course, limitations and threshold because proteins exhibit conserved and diverse regions due to evolutionary constraints and so on. And the real world biological sequences rarely resemble the worst case scenario. And then the greedy incremental clustering, it will also see some problems because the cluster assignment might be impacted by the sequence order. And then if Y is, for example, more similar to B, but it first, the algorithm first hits A during clustering, then Y might end up as part of clustered one. So you will not get the other one as representative of the cluster, but you might get whatever sequence the algorithm is hitting first, basically, right? So those are some problems, but you can still use the CD hit softwares. And then if I quickly ask, let's say this question, so if I use the following commands, following command for CD hit running. So here again, the same parameters that we saw earlier. So can somebody mention what minus C.9 and minus N4 indicate in the chat, perhaps? So minus C.9 was 90% identity as the clustering threshold, and then minus N4 was what size of 4. So 4 was filter being applied, right? So in the clustering concept, Professor Gromiha went over a very important algorithm, essentially, right? So that was called k-means clustering, right? Now, k-means clustering is, of course, like an unsupervised machine learning algorithm, and it is quite widely used as clustering of some data based on different sort of values. So we'll see some examples there as well. Now imagine you have in a 1D array of data. So you have these 12 data points. Okay. So from just simple visualization or some just visually looking at this data, so could someone mention how many clusters or how many groups can we form using this set of data or using this data set? So how many groups of data can we form here? Just visually inspecting it. So of course, you might notice that these 4 data points are present at in at a very close proximity to one another. Excuse me. And these 4 are present again, like kind of grouped together, and these 4 again grouped together, present at very close proximity. So yeah, as I see in the chat. So from visual inspection, you can think that okay. So these data sets, if I group together in 3 clusters, so these will be the representative components or representative members of those clusters, right? So these 4 one, and then these 4 will be another one, and these 4 will be another one. Now, of course, if you ask a computer, a program, how to to differentiate between these data points, so it cannot rely on this visual inspection, right? So it has to resort to some sort of calculations based on which it can say, okay, so these different data points are part of this group, and then the other set of data points are part of these groups, right? So it has to perform some sort of calculations, And one of that is, of course, what we are talking about here, k-means clustering. So here, just mentioned what I just said. So by examining the data, we can easily identify three noticeable clusters, which are these, right? So if we divide into these three colors, so one is the blue color, the green color, and the red color. Fine, right? Now, k-means clustering is the computer's way, you can say, to group data points into cluster for specific number of clusters, right? And of course, for a large data set, so where a lot more number of data are present than 12, so let's say thousands of data are present, so you will definitely need the help of some algorithms to divide them into different groups, and k-means clustering is one of those. Now, first, while running the k-means clustering, you have to identify so how many total number of clusters you want. Now, let's say here we are giving input that we want this data set to be divided into 3 total clusters. So here we have k equals 3. That means 3 total clusters we are intending to get, right? So what the algorithm will do is that from all of the available data points that it has, it will randomly select 3 distinct data points. Okay. So say we have selected through the algorithm these 3 representative data points as the basically the representative member of the different clusters. So blue, green, and red cluster. Next, what the algorithm will do is that it will try to put each of the available or the rest of the available data points to each of these clusters, or to one of these clusters, based on what? Based on distance from the selected clusters, or currently the member or the representative member of the selected clusters. So what are those distances? If you notice, let's say for the first circle that we have here, so based on the distance, let's say, of how far this data point is present from the blue, green, or red one, so based on that, what do you think? Uh, what group these first data point will be part of? Blue group, green group, or red group? So can somebody mention quickly, maybe? Just based on how far the first point is from all of those representative points, so blue, green, and red points. Yeah, so I see one answer, blue. Yeah, so if you consider the distance, let's say, so of course from the blue representative, you will have this distance, right? It's a very small distance. Then from the red cluster or red cluster's representative, you will have this distance. From the red cluster, again, you have a much higher number or much higher value of distance, right? So since blue distance is much smaller, so you can expect that the first data point will be part of the blue cluster, right? Now you have to iterate again this exact process for all of the data points, rest of the data points that are available here, and overall you will find something like this, right? So starting from random selection of a initial, let's say, cluster representatives, ultimately you are getting this sort of distribution for all of the data points available, right? Now, what the algorithm next will do is that now whatever data it has, it will compute the mean position of those particular data points present within a same cluster, right? So, for example, these blue clusters mean will be like this blue pipe here, the blue vertical line here. Then the green clusters representative will be this green vertical line, right? And the red clusters representative will be this red vertical line, because those are the mean points of all the available data within those clusters, right? And next, what the algorithm will do is that it will again iterate through the same process of assigning each data points to one particular cluster. But now, during the distance calculation, it will now count the new representative of the clusters. So basically, the mean point of the earlier cluster groups, right? So again, it will calculate the distance from the new representatives, so the mean points, and then it will find out, okay, again, so for example, the first data point again is part of the blue cluster, right? So it will assign the first data point to be a part of the blue cluster basically, right? And then again it will do the process for the entire thing. And now in this particular case, if you notice that based on, or if you compare with the earlier iteration, so where the step 4 was completed earlier, right? So after that, following computing of the means and then considering that as the representative of the new clusters, and then reassigning each point to the new clusters, right? The new blue, green, and red clusters. So you'll notice that through this iteration, so from step 4 to step 5, through a new iteration, the distribution of the data did not change. So the data points that were considered to be part of the cluster, these 3 different clusters earlier, so in this new iteration also it is staying the same. So now the k-means algorithm will think is that, okay, so if I'm not getting any different result for this cycle, if I'm after certain iterations, so it will consider that, okay, so this cycle is finished, right? So this cycle is finished, and then you have the cluster results as this one, the the marked one here, right? Now, is that an optimal distribution of data? So if you compare earlier from the earlier example of the earlier slide, we used visual comparison, visual inspection to divide into 3 clusters, right? So if you notice in that one, so these 4 were present in a same cluster, these 4 were present in a different cluster, and these 4 were present in a different cluster. But now the k-means algorithm, what it has done is that it has done the grouping differently. Now, if that is optimal or not, for that, the k-means algorithm will have to run again. So the one cycle has completed, but a new cycle has to start, where the algorithm again will consider 3 different random initialization points as part of the representatives of 3 different clusters, right? So the iteration is finished, but this cycle, a new cycle will have to occur basically. And then while that new cycle will finish, you will get a new set of clusters, new set of distribution of data into clusters. And then you have to compare, okay, so earlier cycle we got this result, the new cycle we got this result. So what is basically among these 2 cycles, which one are better? Which one is better as output, right? So based for that, what the algorithm will do is that it will sum up the variation within each cluster. Now, what that essentially means is that, so let's say in this cluster output, from the mean point for the entire distribution of the data, you are getting these variations, right? So variation by which I mean that this length of the horizontal lines, the blue, green, and then the red one. So these will be the variations within each cluster, right? Now, if we add all of this up, the total length of those variations will represent the total variation within the clusters, right? Now, for another cycle, the k-means algorithm will find some new distribution of data points into different clusters. So for example, in this new cycle, basically, it has found the grouping to be slightly different, right? So these 4 are grouped together, then the green is these 4 and the red is these 4 from some other cycle. So where the initial point was random again, right? Different. And here, if you notice the differences and you add the total together, so this is the current iteration's total variation, right? And you will notice that from the earlier iteration, the current iteration has a reduced total variation within the clusters. So the k-means algorithm will think that, okay, because the current here I have written iteration, but you can think of it as current cycles total variation from the earlier cycles essentially. So you will see that whatever or whenever the k-means algorithm will find that, okay, so this one is showing lesser variation, so that means that this is a more optimum distribution of these data points into the clusters, and that is how it basically is done. So after a certain number of cycles, you will see that the k-means algorithm will not be able to again further optimize the distribution of the data into different clusters. So it will think that, okay, so the algorithm is converged, your analysis calculation is converged, and the final output will be the optimal distribution, right? So here, just for illustration purposes, I have given two links. So one link is this YouTube video link, which I have used as the basis for this example. And then another one is the link to an interesting website, uh, which is basically this one, right? Now here in this blog post, what the author has shown uh, is that how the steps of the k-means clustering in assignment and the centroid formation and the new distribution into different clusters and then adjusting the values and etc. works. So firstly, let's say we are picking the initial coordinates by ourselves. So I'll choose here. And then let's say we want to have a Gaussian mixture of data. So we have now these three different sets of data, right? So let's say I'll put three different centroid positions. Now in this one, I can put again outside the data distribution also, that doesn't matter. So let's say one, let's say two, then let's say three. Okay. So I have these three different, okay, let me just do it a little different way. So let's say I have one here and then two here. And then let's say I have three here. Okay. So that's good. Now what I have to do is that I'll just press go here. And then in the first step, if you noticed that the green shaded areas, red shaded areas, and the blue shaded areas, so whichever data points were like falling within those areas, those were assigned to a part of that colored cluster essentially, right? So these shaded areas are essentially called Voronoi representations, and it is basically kind of a distance between these our initial point of clusters, right? So the initial representative cluster points, and then whichever part is coming within that shaded area, it will consider those data points as a part of that cluster essentially. So here the assignment is done in a way that these data points are part of the green cluster, right? Now, if you update the centroid, what will happen is that now the new representative position for these clusters will be to the mean position of the assigned data points for those clusters, right? So it will be something like this. If I updated the position, right? Now you will see that the blue representative has moved in this whole group where the blue data points are present together. The green representative again has moved towards this green group, and then the red representative has again moved this way, right? Then if you reassign the points, now what will happen is that, so you see in the earlier step, these green data points are part of this green shaded area. Now, but now since the centroid of the cluster has moved or the representative has moved, so now these green data points will be part of the red shaded area, right? So now if you reassign, the same thing will happen. Basically, the data points will be now assigned to this red cluster, red shaded area, and then we can update the centroid again. So if you notice that the centroids will move again, so the representative points will move again. And then following the reassignment and update centroids, you will notice that at some point, the whole output that you are getting after certain steps will not change. So now you can say, okay, so these are the optimal distribution for these three different clusters, right? So you can think of it that way. If I start with, let's say, randomly using some sort of uniform points, again, you can just put centroid different number of clusters. You can add centroids here, and then you can try to optimize what would be the distribution based on the k-means algorithm here, right? So that is just for a visual depiction of how this algorithm essentially works. So I will try to again show you another example for the k-means clustering algorithm. So please let me know whether you can see my terminal here. Okay. So hopefully you can see my terminal. I guess it would be better if I just share from the other screen. Just wait a minute. Just let me share the other screen. So here, let's say what I want to cluster the data. So that is this one. From some analysis. So this is uh, a calculation of molecular dynamics trajectory. And what I have gotten is this kind of data set. So we'll see on the left hand column, we have these frames, right? So it is stretching from 1 to 80,000. So total 80,000 different data points are present. And then on the other column, something called mode 1, mode 2, mode 3, mode 4, etc. are visible. So this calculation is basically from something called a dihedral principal component analysis, which is again another grouping technique for, it is also used in molecular dynamics trajectory analysis. So basically using dihedral angles, we are trying to find out uh, the motion of particular region or loop region in a protein based on these different modes. So here these first two modes will essentially represent, because it is a principal component analysis, those will represent the maximum variation within the data set, right? So if we try to cluster this data set using the first two modes, so here the second and third column, you will notice what is the distribution for those two data points is across this data set, right? So for that, I have this scatter plot script in Python. We'll notice that I am loading this data set using the NumPy load text, right? And then because 80,000 is a lot of data points, right? So I'm considering only 10. So I'm just increasing the interval of the data point to be included. So I'm using a step size of 10. That means that every 10th data set will be included. So it will reduce the number of total data set to 8,000, right? And then I have some figure forming commands here, and then some labels and etc. And here I'll just use the scatter plot function, right, to produce a scatter plot. So let's see the distribution of this data. So after running, after this command is run, the Python script, you will notice that the scatter.png file is visible. Okay. So if we see this one clearly, so of course, this is the kind of distribution for mode 1 and mode 2. So mode 1 is on the x-axis where it is written DPC1. Mode 2 is on the y-axis where it is written DPC2. And that is the distribution of the overall data point. Now, of course, we cannot really try to infer, let's say, these data points, like what is the, like if you want to extract certain representative conformations from this data point, we cannot exactly identify, okay, from where we should extract the conformations or which data points will give us best representation of certain types of groupings, right? So for that, what we can do is that we can, of course, rely on the k-means clustering algorithm. So here I have this Python script for k-means clustering, and again the same thing. I am loading the data and then giving a step size of 10. And here, instead of just a plain scatter plot, what I am telling is that the scatter plot will be based on the k-means algorithm. So here, the total number of clusters that I want to divide the data will be 3. And then this initialization algorithm will be k-means plus plus. And then the number of maximum iteration for each cycle, that will be 300. And then the number of initialization, so different number of different times that I will choose an initial representation and then proceed the calculation from there will be 10, right? And then based on these different color, basically color codes, I will use the, I will use that to represent the different clusters, right? So if I run this k-means.py script again, so you'll notice that something was pasted on this terminal. And what was pasted were the basically composition of each of these clusters. So cluster 0, 1, 2, total 3 clusters. As per our command, it has divided the data point into, and the composition of or the composition of members or data points into those clusters in a percentage value it is given, and then the centroid points, the coordinates of those are given. So -1.2, that will be the x coordinate, and then 1.101, that is the y coordinate, and so on, right? So if we notice the clustering plot. So it will look something like this. Okay, so now based on our provided 3 cluster distribution, so based on our provided command in the script, so it has divided the entire data set into 3 different clusters: the orange one, the lime one, and the cyan one, right? And so basically, looking from these colors, you can also see which is the most prominent cluster among these. So can you quickly mention, if I ask you, so which of these colors seem to be the most prominent one, having the highest number of data points? Orange, the green color, lime color, or the cyan color, blue? So you can just take a look at the left-hand side terminal where the composition of each cluster is mentioned, right? So here, as you see on the terminal, what is pasted, that this cluster 2 has 57.66% of data, right? So here, if you look at cluster 2, it is the point minus 2.02, 0.27. So minus 2.02 is somewhere here, right? And then 0.27 is here. So basically, this orange cluster is the most prominent cluster, which has the highest number of population, basically among these 3, right? So if we extract, let's say, structure, a representative structure from this cluster based on these values, so you'll get the most probable orientation of certain loop regions for which the trihetal principal component analysis was conducted. And then the other two will probably give you some slightly lower prominent, but some sort of transition state conformations, right, regarding some proteins. So that is quite useful here. But another concept K-means clustering has is that I have given here the command that it should divide the data points into 3 different clusters, right? So how do I get this 3 different value? The 3 value for which the K-means algorithm will be considering to divide this entire data set into. So that basically will come through some analysis which is called within cluster sum of squares. So what this within cluster sum of squares algorithm will do is that, again, it will try to cluster basically the entire data set into a number of different clusters. So first it will try to cluster the data set into, let's say, 2 clusters, then it will do for 3 clusters, then it will do for 4 clusters, then it will do for 5 clusters, and so on and so forth. And then after each set of optimal clusters for certain cluster numbers it has obtained, so it will try to see, okay, what is the total variation kind of within each cluster for each of these cases, right? And based on that, at a certain point, we will see that the variation is kind of tapered off. So you will see an elbow point present in the analysis. And based on that, you can decide the optimal number of clusters. So if I just plot it quickly. So this is kind of the within cluster sum of square plot. So basically, with each increasing number of cluster, we will see that the WCSS, or within cluster sum of square value, is decreasing, right? So that means that if you want to minimize the within cluster sum of square value, so you can use like a very high number of cluster, points, but that is also not optimal. We will see in a slide there. But what is done basically is that, so at a certain point, you will notice that with increasing number of clusters taken from that point, the within cluster sum of square value is not decreasing by that much amount. So that is basically considered an elbow point. Here the elbow point was kind of present at the number of clusters equals 3. So after that, if you further increase the number of clusters to divide your data into, so it will not reduce the WCSS value by that much. So that means you can consider, okay, so 3 value will be the optimal value for clustering here. So I'll just share my other screen, and basically you can take a look at the within cluster sum of squares. So the within cluster sum of squares is basically done by this formula. So here you have to just take the distance again from the, and it's not working kind of. Anyway, so the within cluster sum of squares value is how you do it is basically is that you have to compute the distance from each data point from its cluster centroid. So here the centroid is considered as like the C1, C2 values and so on. And the individual components are this I, right? Pi here. And then you have to compute that distance and then you have to square that and then you do have summation, right? For all of the different clusters you do, and that will give you the total within cluster sum of squares value or the WCSS value, right? Now, as I discussed, that through the WCSS plot, you have to kind of identify the elbow point. Because if you do not do that, let's say basically, if you do not do that, so you might think that, okay, so if you need to minimize the WCSS value, why not we get a WCSS value of 0? So you can get that if you divide your total data into only that number of clusters. So if you have total of 1,000, a million data points, and if you try to divide that into a million clusters, of course, then each single data point will be considered as each separate cluster, right? So here your WCSS will be 0, the minimum value, right? But does that allow you to infer any sort of significant, uh, any sort of significant, inference regarding the data distribution? And it does not, right? Because it is a mess kind of, right? So considering, of course, the WCSS value as the 0. So if you consider each data point as separate cluster, so that is not helpful. Similarly, it is not helpful to just only get one cluster for all of the data points, which in which case your WCSS will be max. So that is also not good. You have to identify the middle point where the elbow will be forming in this WCSS plot, where after a certain point, further increasing in the number of clusters will not give you that much reduction in the WCSS value. So basically, that is the optimal number of clusters in this case. I have put a link here also. You can check it out maybe. So, so the next concepts were like some other software related concepts where Professor Gomiha went over, for example, other softwares like BlastClust, right? For example, here in this BlastClust input file, basically, so it is mentioned BlastClust -i, so as input in file, -i out file. Then some parameters are mentioned here. So minus p capital T, minus capital L 0.8, minus p capital T, etc. So here the 0.8 stands for what? So in this case, the 0.8 is basically minus L 0.8. So that basically indicates the coverage of 80% between these sequences, right? So here minus L is basically the minimum length coverage. So that is what it is being indicated. So you might take a look into just what kind of parameters we can give to the softwares that Professor Kumiha discussed. Those kinds of questions might come in your assignments or exams, right? So you might take a look at it. Now, another set of concepts that Professor Brahmiha went over in the classes is that, so based on the, considering the amino acid composition between different sequences, we can try to calculate the distance between the two sequences. So how far apart are they present, right? Not in just exactly in space, but how far apart they are. So how much closing characteristics there or how much different characteristics there. So that will kind of sort of give you if those sequences are related or not, right? So that is kind of where that is kind of the concept where the distance analysis is coming into place. So here, if I take a look at one of the distance calculations, that was Hamming distance calculations using amino acid composition. So if you have these two sequences, what you can do is that you can calculate the composition calculate the composition for a particular amino acid, let's say alanine, for sequence 1 and sequence 2 also. Then you can subtract that. So you will basically get a composition difference. You take an absolute value of that And for all of the amino acids present, so the total 20 amino acids that are present, for all of that, you try to calculate these composition differences. And ultimately, you add all of that up, and that will give you a distance, basically, right? How, in terms of composition, how closely related these sequences are, or how far apart not related these sequences are, basically. So if we take a look at, for example, this one, so all of these amino acids I have listed here, right? So the total 20 amino acids are here. Let's say for alanine. So how many alanine is in the first sequence? First sequence here. So we have 1 here, 2 here, 3 here, right? So total 3 divided by 10 because it is 10 length, right? So 3 divided by 10 is 0.3. Let's say we consider that as composition. So 0.3 for sequence 1 for alanine. And then for alanine in sequence 2, it will be uh, let's say we just go through all of this. So alanine for sequence 2, it will be again 0.3 because again in sequence 2 also 3 alanines are present, right? At different positions, but the overall composition is same for these sequences. So here you will get composition 1 minus composition 2 for alanine as 0, right? Similarly, if you calculate for aspartate, so how many aspartate was there? 1 aspartate out of 10. In sequence 2 also 1 aspartate in the total of 10. So you'll get 0.1. So 0.1 minus 0.1 will be 0. Will be 0, right? And then in some cases, you will notice, you might notice basically that there is a difference. So in that case, that subtraction and their absolute value will give you some sort of value, right? But here, of course, as we see here, the total differences for the amino acids that are present, for each of them it was 0. So sum that up will give you a total of 0. So here, using amino acid composition, the Hamming distance will be 0. Okay. So that was kind of the concept. We might take a look at another example where in some amino acid the composition values are different. So that will give you some sort of value regarding the Hamming distance. First, can we try to calculate, let's say, the sequence identity problem? So if you have been given two different sequences and you are asked, okay, try to calculate the identity between these two. So can you maybe do it? Uh, Okay, I have just given the formula here. So it's very simple. It's basically the number of identical residues that is present, and then you divide that by the length of the shorter sequence. So let's say the length of these two sequences are different. So one was like 2 length higher, right? One had 2 more amino acid residues present there. But you would consider the length of the shorter sequences, which you will use to divide these identical residue numbers, right? So in this case, since 8 identical residues were there, only 2 places we are seeing differences. So S versus T is a mismatch, M versus R is a mismatch. The rest are same. So 8 divided by 10 is 80%. So that means sequence identity is 80%, right? So these are some simple numeric calculations that you might face in the exams. So can you try to calculate the Hamming distance between these two sequences? So it's similar calculation. You have to just calculate the composition for the amino acids, individual amino acids, and then you subtract the composition one from another, and then you just take an absolute value and sum it up. So can we try to calculate this one? So take a moment, just calculate it and share in the chat. So you have to again follow the same procedure. So for example, for alanine, how many are present in the first one? We have 2, right? And you will see like 0.2 for the composition, right? For the second one, okay, so if we place all of the values here, yeah. So for the second one, you will see 0.3 in the composition, right, for alanine. Of course, there is a difference. So 0.2 minus 0.3 or vice versa, you will keep an absolute value of 0.1. So you have to consider that. Then for histidine, it is same. For isoleucine, it is again you are getting 0.1 difference. Lysine same, leucine 0.1 again. For tryptophan again, it is 0.1. So if you sum all of that up, ultimately you are getting 4 0.1s from here. And then you get the total value of Hamming distance as 0.4, right? So these sort of Hamming distance calculation, etc., you might face in the assignments also. So very simple formula, just keep in mind. You have to calculate the composition for each individual amino acids across a set of sequences and then compute the difference, right? And then sum it up. Now, another concept that Professor Brumia again in the same category. So he went over the Euclidean distance, right? So here, for example, if I have two sequences, now the concept is kind of same. We have to calculate the distance again based on composition. But here what you will do is that earlier cases you saw that comp1 minus comp2 and you took an absolute value of that difference, right? But here what you will do is that you will first make a square of that comp1 minus comp2 for each individual amino acids. And then after you do the summation, you just take a root over value, square root value of that, right? So it is slightly different approach here. So if you can calculate for all of the values here, you will see in some cases you will see difference. For example, in alanine you see 0.1 difference. For aspartate, it was not present in sequence 1, but it was having a 0.1 value in sequence 2. So again, the difference is 0.1 here. For isoleucine, you have 0.1. Then the lysine is same. Leucine is 0.1. Serine is also 0.1. So there is 1, 2, 3, 4, 5. Okay, oh, sorry. The aspartate was actually present. It was 0.1 again. So you have basically difference of in 4 places, right? In alanine case, in isoleucine's case, leucine's case, and the serine's case. Okay. So if you just square all of these values, 4 0.1, you will get 4 0.01s basically after squaring, right? Sum all of that up, you will get 0.04, and then you take a square root, you will get 0.2. So that is the calculation for Euclidean distance. Slightly different than the Hamming distance. You just square up the differences, and then at the end, after summation, you take a square. Okay. So just keep this formula in mind. Okay. So if we come to the next question, so let's say you are asked some sort of reasoning type of question. So for the following conditions, which one represents a set of non-redundant sequences? So the best representation for non-redundant sequences. So can somebody quickly answer in the chat? So 4 options. Whether the two sequences should have similarity of less than 75, similarity of greater than 80%, no two sequences, or no two sequences have similarity greater than 15%, or no two sequences have a similarity of less than 20%. So what do you think will the answer be here? Mention in the chat. I see, I think option C answer. Yeah, exactly. So no two sequences should have similarity of greater than 15%. So that means the sequence similarity will always be less than 15%. So that means they will be non-redundant, right? So the other options kind of fall short at that part. So if you carefully look at it. So these sort of reasoning type of questions might again come in the assignments. So another type of kind of calculations that we can do is that since we are talking about secondary structure formation and so on. So for secondary structures, which amino acids seem to be more prominent or more important in certain type of secondary structure formations than others, right? So that is the sort of inference that you can obtain from this type of calculations. For example, here it is asking that, okay, so what is the propensity of glycine to be in alpha helix? So if it has a high propensity to be present in alpha helix, then you might think, okay, it has a high propensity. So if I say like a number of glycines, so it could be part of the secondary structure could be alpha helical in that case, right? So those sort of inferences you might draw. And here basically what you need is that you need to calculate the percentage of a certain type of residue. So residue I in helix, you have to calculate that. So it will be n alpha I divided by ni, where n alpha I is the type I residue, how many of that are present in helix, and then all of the type I residue, how many of that are present in the whole data set. So for example, if you look at here, so type I in this case is glycine. So how many of them are present in helix? I can see, I think here, one glycine corresponding to H helix, right? So one glycine is present in helix, and in the entire data set, 1, 2, and 3 glycines are present. So the this value, I think I will get 1 by 3 percentage. So times 100 if I have to take kind of. But anyway, it will get crossed out, I guess, in the final result. You have to do kind of this calculation. So n alpha pi divided by ni. Now you have to divide that by n alpha by n. So what is the n alpha by n? Here the n alpha will be the total number of residues present in helix. Divide that by total number of residues in the whole data set. So this is kind of a normalization kind of calculation, I guess you can say. So you have to normalize by the percentage of all residues in helix. So the upper, so here you got like 1 by 3 for the n alpha I by ni, and then here you got 5 by 10 for the n alpha by n. If you compute that, you will get 0.67. So the propensity of glycine to be present in alpha helix will be 0.67, right? So you can use the formula here to do this kind of calculation, right? Now, based on the propensity values, for example, here you calculated 0.67, you can infer some sort of significance of that, right? So there is another method that is called Chow-Fastman method. I think it was discussed by Professor Bromia. So based on that method, calculating propensities and so on. So alanine and tyrosine can be represented as the best way for which of the following options? Can you mention quickly in the chat maybe if you remember the Chow-Fastman method? So here basically it is trying to say that alpha helix format will mean that at alpha C helices that residue will have a very high prominence, right? A very high propensity. And then beta sheet format again, beta strand format. So at beta strands, that particular type of residue will have very high propensity. Then if it is mentioned as breaker, so that means that it essentially means that for that particular secondary structure, for example, here in beta strand, if it is a breaker, that means it will have much low propensity. So essentially indicates that, okay, so if that residue was somehow introduced here, so it would probably break the secondary structure, right? So in the Chow-Fastman method, what is kind of the calculated propensity values here? So here you have different values, right? For alpha helix, beta strand, and turns also, it is present here. For alanine and tyrosine, if you notice, so for alanine, the alpha helix corresponding value is 1.42. For tyrosine, the beta strand corresponding value is 1.47. Now, the interpretation of this is that if it is higher than 1 for this particular method, so if it is higher than 1, then that will be a former. So that means that residue has high propensity in that type of secondary structures. If it is lower than 1, so that means it is a breaker, or conversely, that is present at a much less propensity value in that type of secondary structures, right? So for both alanine and alpha C helix and tyrosine and beta strand, it is having 1.47, so a very high value, well over 1. So that is these are formers, right, for the corresponding secondary structures that you mentioned. So can anybody mention, let's say from this list, so aspartic acid, whether that will be alpha C helix former or breaker, and whether that will be beta strand former or breaker for aspartic acid. So if you notice quickly here, for aspartic acid, it seems that in the alpha C helix value, it is 1.01. So yeah, it is kind of over 1, but it is not exactly a very strong format. where it has like 1.3, 1.4, 1.5 kind of value, kind of at the at the neutral point here. But for beta strand, it is 0.54, so that is very strong breaker. So if an aspartate was present in a beta strand, it will most likely break the beta strand. Or conversely, you can say that aspartate is not very prominent residue to be present in a beta strand structure, right? So that kind of way you can interpret this table here. Okay. So I think we will wrap up the class today. So I think I've discussed most of the concepts, I guess, from the other class. Yeah. So total in all, we discussed the clustering algorithms, the softwares, for example, CD hit, and some concepts about the secondary structures and so on. And then ultimately some calculations based on amino acid compositions, which will let us know whether basically these different sequences are similar or not similar in the composition kind of way, right? From the perspective of compositions. Okay, so I think I will wrap up today's class. So thank you everyone for joining. Again, we will see in the next class. So yeah, so please try to also attend the other TA sessions as well. From on Saturdays and Mondays, and try to carefully follow Professor Govia's lectures. So yeah.
Thank you so much.
Yeah. Thank you. So again, we will continue with the further tutorial classes. Thank you.